Thank you, Dr. Agrawal. This is a very nice introduction. First of all, I want to thank you for your, generous invitation for me to give a talk on this to this very prestigious group, and it's definitely for the Saturday. I think this is a great start for me. And again, as I said, the COVID crisis is such a bad in India. And my heart goes out to all of you and your family members, and I hope all of you have a strength to come out of this very soon. So with that, I will start sharing my slide, and then I will start my talk. So as you said, my topic will be on immunotherapy in multiple myeloma. The reason I chose this topic is because this has been a hot topic for several cancers, and in particular myeloma as well. So we'll see what is the development that has happened in multiple myeloma in this aspect, and what are the things that we have been doing here around this, the immunotherapy and the translational aspect of immunotherapy in myeloma. These are some of my disclosures. I will be talking about some of the non-FDA approved uses as well. And there are three learning objectives that I will talk. One is to understand the immune regulation of multiple myeloma. Next is to describe the immunotherapeutic options in multiple myeloma and the promise that it has. And lastly, to discuss the potential challenges that are associated with myeloma immunotherapy, and how could we overcome those challenges for a very effective delivery of the immunotherapy to our patients. Now, this is a very famous kind of a paper from Chen in 2013 that looked at what we call the cancer immunity cycle. So for an anti-cancer immune response to lead to very effective killing of cancer cells, a series of stepwise events must be initiated, and that need to be allowed to proceed and expand iteratively. So there are mainly 7 steps that need to happen for the immune cells to lead to effective anti-cancer killing. There are 7 steps that are outlined here. So starting from the recognition of the antigens. Once you have the cancer cells die, they will release new antigens, and that will be captured by what we call it the antigen presenting cell, or in particular the dendritic cell, and they lead to the priming of the T cells, and the T cells are now trafficked to the tumor site, and in the tumor site they're recognized by the T cell receptor, and ultimately leads to the killing of these cancer cells. This is a must. Now, how is myeloma in terms of the potential role of immunotherapy? So this is another very famous paper that looked at the prevalence of somatic mutations in different cancers. So the dots here represents the sample. The red horizontal line represents the median number of mutations per sample. And the axis, the horizontal axis looks at the different cancers, and the vertical axis looks at the somatic mutations prevalence per megabase of that cancer cell. As you can see, a myeloma occupies the middle of a range of that position, suggesting that the potential of the immunotherapy can be applied in myeloma because of the prevalence of the somatic mutations. It's definitely not high like squamous cell cancer or melanoma, which that is a very highly immunogenic tumor, but also it's not very low like some of the brain cancers like medulloblastoma or astrocytoma. So it occupies in the middle and suggesting there is a potential role of immunotherapy in cancer. Now, the next is what is the evolution of the myeloma over time. That also tells us the potential role of immune targeted options in this disease. So this is the evolution of the myeloma from the normal cell. As we all know, this starts from MGUS, goes to small ring multiple myeloma, then multiple myeloma, and then the other high risk states. As you can see that there is an impact of both genomic and the immune changes that need to happen for these states to evolve from very precursor stage to very advanced stage. In particular, there is a balance. There needs to be kind of a contribution from both tumor promoting and tumor suppressing immune cells in combination with the both primary and secondary immune change and the genetic changes, and that lead to the emergence of this, the evolution of this disease. So looking at this, you can kind of envisage that potential role of immune options in targeting these different cellular cells that contribute to the development of the disease. And lastly, if you look at the microenvironment of the myeloma, so myeloma is a kind of a niche-dependent disease. There is interplay between the infiltrating myeloid cells, tumor suppressor cells, the tumor cell, and more importantly, extracellular matrix, which I'm going to talk about a little bit later. So both had to combine, and then they kind of interact with each other and create a very immunosuppressive microenvironment that contribute, or that provides a very perfect environment for the tumor to kind of myeloma cells to kind of evolve. And this is what we call as the immunostatin multiple myeloma, or immunostatin microenvironment. So the microenvironment is very immunosuppressive. In particular, as shown here in the figure, there has to be, there are either immunogenic cells who try to help control the cancer, and there are tolerogenic cells, in particular the myeloid-derived suppressor cells, the M2 type of macrophages, the extracellular matrix, and even the dysfunctional antigen-presenting cells. So we know that in myeloma, there is a predominance of this immunosuppressive cells. In particular, there is dysfunctional dendritic cells. There is a high kind of concentration of the tumor-associated macrophages, the myeloid-derived suppressor cells, and some of the cytokines, and leading to the exhaustion or the energy of the T cells. So this kind of sets the stage, I would say, to target, to kind of redress this kind of different pathways and potentially use the different immune options in multiple myeloma. So we have a very strong preclinical rationale and biological rationale to use the immunotherapy in multiple myeloma. Now, what we know so far in terms of the potential role of immunotherapy in multiple myeloma. As you know, myeloma, we have enough evidence from the allogeneic stem cell transplant for the role of immunotherapy, number one. Number two, we saw in those patients who received allogeneic stem cell transplant, those who had a chronic GVHD, graft versus host disease, their survival was better than the people who did not have a graft versus host disease, chronic graft versus host disease. And lastly, those patients who received allogeneic transplant, you can salvage, you can even control the myeloma in some of the cases by using donor lymphocyte infusions. So that kind of makes a case for immunotherapy. Now we have enough evidence for the role of immunotherapy with the use of monoclonal antibodies, the derivatives of monoclonal antibodies, and last but not the least, the cellular immunotherapies, which is kind of taking a center stage in multiple myeloma right now. Now, looking back to the cancer immunity cycle, the different emerging targets and the established targets are kind of being targeted at different steps. In particular, for the step one, there is a targeted therapy for the antigens, drugs like monoclonal antibodies, which I'm going to talk about. And in terms of the antigen presenting stage, we have some data on vaccines in myeloma, the tumor vaccines, and which I'll briefly explain that. And then we have using the direct kind of active immunotherapy using the T cell modulation. We have enough evidence now with the use of cellular immunotherapy, in particular CAR T cell therapy, and also using the bystanding T cell with the bispecific antibodies. And lastly, to reverse the immunostat, or to reverse that immune-mediated paralysis in the microenvironment, we have evidence of checkpoints, though we have, we'll talk about the bumpy start that we have with checkpoints, but more importantly, we have enough evidence from the immunomodulated drugs to reverse that immune paralysis. So these established and emerging targets are attacking or using one of these pathways that is, used in the cancer immunity cycle. Now, in conceptually, there are three principles, three broad categories of immune options in multiple myeloma. One is that the agents that reverse the tumor-mediated immune paralysis, drugs like immunomodulated drugs and immune checkpoint inhibitors. So that helps to reverse or augment the immune system to fight against the cancer. Number two is the direct targets, directly attacks the malignant clone, in particular with the use of monoclonal antibodies. And we have enough evidence of the use of drugs like daratumumab, isatuximab, elotuzumab that kind of help in control the disease. And last but not the least, the use of active immunotherapy using engineered CAR T cell, the dendritic cell, and other active cellular therapies like NK CAR and all those stuff in development. And so these are in this category. So mainly there are three kind of principles or three categories that we can conceptually or broadly kind of classify these immune options in multiple myeloma. Now, going to the monoclonal antibodies, that the agents that directly target the myeloma cells. So we know the development of monoclonal antibodies, in particular the CD38 monoclonal antibodies, is considered a breakthrough event in multiple myeloma therapeutics. In addition to this CD38 antibody daratumumab, we have another CD38 antibody called isatuximab, which was FDA approved last year. And we also have another the monoclonal antibody against CS1 or SLAMF7, the elotuzumab, and we have enough data from that. The mechanism of action of these monoclonal antibodies is as shown here. This is either they can directly kill the tumor cells, or they can kill the tumor cells through antibody-dependent cellular cytotoxicity, or they can also kill through the complement-mediated pathway. The other excitement that is happening in the platform of monoclonal antibodies and with the derivatives is a drug called antibody-drug conjugate, which is basically a monoclonal antibody with the payload. And in myeloma, that compound is just recently approved is belantamab mafodotin or belamab, which is a target against the BCMA, and this carries a payload called MMAF, and that kills the tumor cells. And lastly is the bispecific antibodies, or what we call as BiTE. There are several BiTEs in development right now, and this is the mechanism of action of the BiTE. They target to two different targets, one to the tumor cell and the other one to the T cell, and it brings this bystanding T cell in close proximity to the tumor cell and leading to the killing of these tumor cells. And these are some of the BiTEs. Mainly there are BCMA BiTEs, and now we have several other non-BCMA BiTEs as well. So if you look at the development of this the immunotherapy, one of the antigen or one of the target that is being used in most of the cases is this antigen called B cell maturation antigen or BCMA. So this has been considered a novel target for myeloma immunotherapy. So a few words on the BCMA. What is BCMA? It's a TNF receptor superfamily of kind of receptor. It is present in long-lived plasma cells or the mature plasma cells, and it leads to the survival of this long-lived plasma cell. The two main ligands are APRIL and BAF. They are the ligands for the BCMA. And in addition to the myeloma cells, it also, in addition to the long-lived plasma cell, it also leads to the proliferation and survival of myeloma cells. In addition, it also helps in create immunosuppressive microenvironment that I mentioned to you before, and it is set by an enzyme called gamma secretase. So there are a lot of trials right now trying to increase the expression of the BCMA using the inhibitor of the gamma secretase. And in addition to the BCMA, there are two other receptors called TACI and BAFF-R receptor that leads, all of them contribute or kind of working together as shown in the figure in help in the growth and development of these myeloma cells. So right now, the BCMA is the lead antigen target for multiple myeloma. Having said that, there are several other antigens being explored and being used in combination with BCMA as well. Now, a few words on the antibody-drug conjugate in multiple myeloma. On the right-hand side of your figure, this is a figure that explains the mechanism of action of the antibody-drug conjugate. In particular, this is for belantamab mafodotin or belamab, which targets the BCMA as a lead antigen target, and then it has a payload called MMAF, which disrupts the microtubule. So it attaches the target with through the BCMA, and then during the process it releases the MMAF, and that leads to the microtubule inhibition and leading to the cell death. Now the phase 1 study of this compound was presented a few years ago that led to a very promising efficacy of this drug as a single agent with the dose escalation and expansion phase, and that led to about 60% response rate, and leading to the design of the phase 2 pivotal phase 2 trial, which is called DREAMM-2 trial. And the key efficacy and safety data is presented here. There are two doses were tested in the DREAMM-2 trial. One is 2.5 milligram per kg, and the other one is 3.4 milligram per kg. Here is the results from the 2.5 milligram per kg. There were 97 patients who were treated in that particular dose. As you can see, it's pretty heavily treated patients. About more than 4 lines of therapy were used in 84% of the patients. There are two main safety concerns. One is cytopenias. The grade 3 and 4 thrombocytopenia and anemia were present in about 20% of the cases each. But more importantly, the main side effect of concern is keratopathy or corneal events. And in that, the keratopathy of grade 3-4 was present in 27% of the patients. The overall response rate was 31% for heavily treated patients, as shown here. The median progression-free survival was about 2.8 months. It's not that very long, but if you look at the median duration of response, In the patients who did respond to this agent, their median duration of response was more than 10 months. So the FDA, based on this data, led to the approval of this agent in August of 2020. One of the main concerns, because of the keratopathy, the patient who needs to be enrolled has to be enrolled in a REMS program where these patients need to have an established eye doctor. You need to make sure that the patient see the eye doctor before you dose the patient at the baseline, and with each subsequent dose to make sure the patient cornea is fine, and that is a defined guidelines on how to adjust the dose and decrease the dose based on the corneal events. Right now, this compound is being investigated in combination with several other agents like pomalidomide, Velcade, and so on and so forth in different stages of the multiple myeloma. But this, as a single agent with this, response rate in these heavily pretreated patients is pretty exciting. Now, the other compound in the same class was presented at ASH and last year was MEDI2228. This is another ADC in multiple myeloma. The mechanism of action is pretty much similar to what we talked about, with the exception that the payload is different. The payload in this case is pyrrolobenzodiazepine, and this is was tested in dose escalation and expansion phase. This is some of the key data. There were 82 patients treated. The median age was 69. They're also very heavily treated. As you can see, about 20% plus patients were more than 8 prior lines. Again, looking at the grade 3 for thrombocytopenia was similar to belantamab mafodotin. Interestingly, this patient didn't develop any keratopathy, but they did have some photophobia, which seems to be a class effect. About 13.4% of the patients have grade 3 for photophobia, but no keratopathy. And the overall response rate is similar to what you saw in belantamab mafodotin, the phase 1 trial, which is roughly about 61% in this heavily treated. So about 61% as well. It will be nice to see the further development of this compound, in particular, sparing the keratopathy in this patient. Unfortunately, from what I heard, that this compound is not going further in the development, so which is kind of a sad news. Now, let's talk about the bispecific antibodies. So bispecific antibodies, several thought leaders now think this is another revolution in myeloma immunotherapy because of several reasons. So the middle here gives the mechanism of action, which I already explained to you before. The lead target again here is BCMA for majority of the bispecific antibodies, and it brings in close proximity to the T cell and the tumor cell, and helps in the killing of the cancer cell with the help of the T cells. So the first in-human BiTE was first presented at ASH in 2018. This is from the Amgen BiTE, Amgen 420, and this BiTE was studied in the phase 1 dose escalation fashion in heavily treated patients. And one of the drawback of this BiTE is it was a short-acting BiTE, so the patient needed to have 2 weeks of continuous infusion, and they enrolled about 42 patients. And this is some of the baseline data. The median prior lines of therapy was 4. And this is some of the side effect. The main side effect was CRS and some infections, but interestingly, there was some peripheral neuropathy was seen as well. The maximum tolerated dose was 400 microgram per day. And this was for that MTD for 10 patients, the response rate was pretty impressive for the type of patients they enrolled. And the right here shows those patients who have deep response continue to have MRD negative responses. And after this, there are several other BiTEs being developed in multiple myeloma. Actually, Amgen stopped the production of this 420 BiTE, and they went into Amgen 701 BiTE, which is a long-acting BiTE and is weekly infusion. And from till that time, now we have several other BiTEs. In addition to the BCMA BiTE, we have now a non-BCMA BiTE, which is one is GPRC5D, G protein coupled receptor 5D BiTE from Janssen, which is also called as talquetamab. And so GPRC5D is a target present in multiple myeloma. It is expressed very limited in healthy human tissue. It is mainly present in primary myeloma cells and also present in hair follicles. It is highly expressed in myeloma cells, in particular those patients with the poor prognostic features of multiple myeloma. And as compared to the BCMA, there is no extracellular setting of this agent. That is one target that is being explored for BiTE. The other target is FcRH5, which is a Fc receptor homolog 5. Again, it is expressed in myeloma cells with almost 100% prevalence. It is also expressed in B cells like CD38 and even BCMA, but is higher in myeloma and plasma cells. It is present in chromosome 1, and Genentech has developed this BiTE against the FcRH5 with the CD3 called cevostamab, and they had the data presented at ASH last year as well. Now, the summary. So now we have a kind of a explosion of BiTE in multiple myeloma with the targets mainly on BCMA, but also have non-BCMA targets. And this is some of the key BiTEs that the summary of the key BiTEs that were presented at last year. None of them have been approved yet. Hopefully soon we'll see the approval of one of these BiTEs, in particular the BCMA BiTE. The top 4 ones are the BCMA, and the last 2 are the non-BCMA BiTEs. As you can see, the prior lines of the type of patients they were enrolled in these studies were pretty comparable, were the median prior lines of 6+. The dosing is different. For example, teclistamab, which is a BCMA BiTE from Janssen, already coming with sub-Q, which is, of course, it's much easier when you try to decide which BiTE to give to a patient. And talquetamab is another one from BiTE, and one from Janssen. The GPRC5D BiTE is also coming as sub-Q. The dosing frequency is also different. Most of them are weekly, but some are Q3 weeks BiTE as well. The overall response rate, I would say, is pretty comparable at the stage. For phase 1, the ballpark of 50 to 80%, depending on the dose. There are the side effects of CRS, the cytokine release syndrome, and neurotoxicity is there, but definitely much less than chimeric antigen receptor T-cell therapy or CAR T-cell therapy. So it is exciting stage in the BiTE space in multiple myeloma, especially with the development of other non-BCMA BiTEs, because as we know, the BCMA being a very crowded space in myeloma already, and so we need some optional or other BiTEs that could be effective in non-BCMA or BCMA relapses as well. Now, if you summarize this, the monoclonal antibodies and the derivatives, I think this is what I can summarize. They are off the shelf. That is the biggest advantage, off the shelf. Given the nature of the disease, sometimes you can't wait for the patients to wait longer to provide any effective therapy. So you have to kind of make a very quick decision and administer the drug. And in that setting, all these monoclonal antibodies, bispecific antibodies, or ADCs have the off-the-shelf kind of advantage. Definitely, in particular, the bispecific antibodies have a very promising early results, and we have to see how that pans out in the pivotal trials and other like phase 3 trials. But most important thing, I think, in addition to the off-the-shelf, is they have a single agent activity. So having a single agent activity is very, very important. For example, the BCMA ADC, the belantamab mafodotin, is a single agent giving a response rate of 31% without a steroid. It's a pretty important thing for patients who always have to bear the side effect of the long-term steroid use. But last but not the least, I think at this stage, we have to see how this pans out in terms of sequencing and where it really fits in the myeloma, and what is the rational combination that needs to be optimized, needs to be seen in further trials. But I would say all these 3 agents are very significant development immunotherapeutic options in multiple myeloma. Now, a few words on the status of the T cell in myeloma microenvironment. So when you look at the T cells, the T cells are the main kind of a very important cell in controlling the cancer, not only in the myeloma but also in other cancers as well. So the tumor progress despite the T cells. The antigen tumor antigen associated specific T cell are present, but the tumor progress despite that, and that is mainly reason because there is a failure of the T cells, and there are several mechanisms that contribute to the failure of the T cells, and these are the different T cell states as outlined here in the figure. They could be anergic or exhausted or senescent T cells, and the goal is to get them more towards the stem kind of promote or introduce the more stemness kind of characteristics to treat the patients with the cancer. And there are the different states of the T cells are marked by different kind of markers as shown here. For example, the exhausted T cell, which is lack of response to the effector response, characterized by lack the expression of the checkpoints, PD-1, TIM3, LAG3, and so on and so forth. Similarly, the senescent T cells are result of aging or chronic infection of the chronic stimulation, and so on and so forth. So what we know is the T cells are dysfunctional in multiple myeloma, whether it's exhaustion or senescence. There is a kind of debate there, but it's a component of probably both. Those T cells are dysfunctional in multiple myeloma. So what that brings up is the rationale for using an agent to reverse that T cell exhaustion, or to reverse the kind of the breaks that kind of put the T cells malfunction in multiple myeloma, or in particular use of checkpoints inhibitor in multiple myeloma. So where we are right now in the use of checkpoints in multiple myeloma. Unfortunately, the PD-1 story is one of the checkpoint against the PD-1, which is, as you all know, is being very widely used in other cancers. The PD-1 story in myeloma has been a sobering reality. This is a summary of the 3 trials that were used the PD-1 agent in multiple myeloma in combination with IMiDs, the Keynote 183, Keynote 185, and Checkmate 602. The agent, whatever was used in this in the trial, were given here, in particular the pembrolizumab and nivolumab. And unfortunately, the patient in the experimental arm, or the patient who received this PD-1 in combination with the IMiD, they had worse outcome, worse survival, worse side effects, and some single agent, some activity is there in terms of overall response rate, but their overall survival for both overall, the overall survival and progressive survival was worse in the experimental arm. Now, what we saw in this is the common denominator in all this was IMiD. So that explains, or that kind of brings up the question is this because of the partner that is causing more side effect and more deaths? Because as a single agent, there is no single agent activity of PD-1, and we saw that in the prior studies where they looked at all the patients with different hematological cancers. Unfortunately, myeloma had no in myeloma that there is no single agent activity, but in combined with the IMiD, there was activity. Unfortunately, that led to the higher side effect. So that studies were put in clinical hold by FDA, and so that got stopped now. Now the question is this the end of the PD-1 story or the checkpoint story in multiple myeloma? And that is the question that is ongoing right now and being investigated. But I think one of the questions that brings up is maybe we probably need a optimal combination or novel checkpoints that we need. So what we have so far in terms of the novel checkpoints in myeloma. One of the things, before I go there, is this is some of the cause of deaths that happen in the PD-1 clinical trials in myeloma, not due to disease progression. As you can see, all of this mainly related to the immune-related adverse event leading to the death of these patients. In particular, the combination of the PD-1 with the IMiD lead to the excessive of the immune-related adverse event. So to answer that question, the book is closed for checkpoint inhibitors in myeloma. There are the novel checkpoints are needed, and one of the novel checkpoints beyond PD-1 that is being investigated right now is a TIGIT. So the TIGIT stands for T cell immunoglobulin and ITIM domains, and which is the immune checkpoints present in both T cells and NK cells, and it competes with another stimulator called CD226, or also called a DNAM. So DNAM is used in immune surveillance in multiple myeloma, and that from preclinical model that we know. And the TIGIT competes with that. So there is interest in exploring this TIGIT pathway, and then the preclinical data we have is 2 very strong papers that came from a group from Australia looking at the role of TIGIT in myeloma. And both are preclinical paper, the mouse model. The first one is looking at from the Guleri et al. Is the looking at the response, the CD8 T cell response in patients with in mouse with myeloma. And what they found out is that in myeloma, if the myeloma burden is higher, the myeloma T cells, the CD8 T cells are exhausted with the higher expression of the TIGIT here. And when they combine with the anti-TIGIT antibody, the myeloma burden went down. And the next one is they looked at the condition of relapse after transplant. So in the mouse model, there are 2 groups of mouse. One is control myeloma after transplant. Other one is relapsed myeloma. And when they looked at the relapsed myeloma, they found out those CD8 T plus cells are heavily expressed on TIGIT antibody. And that is mainly mediated by a marker called IL-10, secreted by the myeloid cells. So there is an interaction of the myeloid cells, what brings back to the same question before, the contribution of the microenvironment from the myeloid derived suppressor cells and tumor-associated macrophages. So that secreting the IL-10, leading to the exhaustion of the T cells, leading to the higher TIGIT expression. So both this paper kind of underscores the importance of the TIGIT as a potential checkpoint, novel checkpoint in multiple myeloma. And there are now several clinical trials going on right now with the use of TIGIT by itself and also in combination with the PD-1. We have a trial which is starting to enroll the patient soon. And also there is another checkpoint which I explained before, is a LAG3 that also is being explored in multiple myeloma. So TIGIT by itself in combination with the PD-1 in combination with LAG3 is being investigated in different clinical trials. So we have to see whether the checkpoints, the novel checkpoints, holds promise in multiple myeloma or not. So in summary, what we can say about checkpoints is checkpoints are active in myeloma. We know that based on the PD-1, based on the TIGIT and LAG3. Mechanistically, I think IMiD is a better partner because they help also in the Ikaros checkpoint release, help in the better efficacy of the IMiD as well, because they depend on Ikaros and ILOs for their activity. But the problem is, of course, the safety, especially when they combine together. Maybe we need better patient selection. Maybe we need better optimal combination. For example, maybe use of checkpoints in combination with the vaccines or in combination with the CAR-T, because ultimately the CAR-T can be exhausted and you need to reverse that kind of exhaustion. And could be a rate limiting step for both of these immunotherapies, both for the CAR-T and the vaccines. But more importantly, we have to see how the novel checkpoints, TIGIT and LAG3, which is being investigated in clinical trials, will fare in the future. But at this point in time, I would say that the TIGIT story is, the checkpoint story is not yet done in multiple myeloma. What about the vaccine-based approach? So there is a lot of interest in tumor vaccines, and myeloma is not an exception. So the underlying principle is to disrupt the tumor-associated tolerance and to activate the tumor-specific lymphocytes. Now, you present the enhanced tumor epitopes in the context of immune activation to the dendritic cells. And this is being explored in myeloma by a group, mainly by several groups. One of the limitations is what is the antigen that you want to choose, and the other one, the rate limiting step, we talked about the immunosuppressive milieu that happens in myeloma. And so there are several points that need to be considered when you talk about the vaccine-based approach, not only myeloma, but for any other cancer for that matter. The optimal antigenic target, what is the platform, what kind of adjuvants that you're going to use, what is the appropriate setting, and what are the immunomodulator approaches that you can combine for the increasing the efficacy of the vaccine. Now, one of the important cell that needs to be responsible for the vaccine, tumor vaccine, is antigen-presenting cell. But in myeloma, we know that the antigen-presenting cells are quite dysfunctional, and based on the several studies. So the David Evican group from Harvard, they looked at combining the antigen-presenting cell, the dendritic cell, in combination with the myeloma cell, the fused together, and to develop a vaccine. And the idea is to present a broad array of antigens, not only this particular antigen, but broad array of antigens in the context of DC-mediated co-stimulation. So they used this platform and they did the phase 1 study. And this is the results of the phase 1 trial that was published in Blood in 2011. There were about decent number of very heavily treated patients. The median lines of treatment were 4, and then about 14 patients had prior autologous transplant, and they tried 3 different doses of the vaccine, from 1, 2, and 4. And this is some of the responses. They found all the vaccine patients had any major side effects, except for some local reaction. And when they looked at the response, they are also able to see that the patients who had vaccine had about 10-fold expansion of the tumor-reactive lymphocytes, both CD4 and CD8. So this kind of suggested that the potential role of vaccine in tumor vaccine in myeloma immunotherapy, and led to the design of this trial called BMT-CTN 1401 trial, which is DC myeloma vaccine study, and which we are a part of. We enroll maximum number of patients in this trial. Is a phase 2 randomized study, and then the vaccine is used after transplant. So in that time when the the immunomodulatory cells, or mainly the immunosuppressive cells, are at the very low level, and they are randomized into 3 arms. It's a 2:1:1 randomization. One arm gets vaccination plus GM-CSF plus lenalidomide, and the other 2 arms get either len-GM-CSF or len alone. So about 230 patients were enrolled in the study and randomized, and we have already completed the accrual of the study, and we're waiting for the results. Hopefully by this ASH, we should be able to get the result out to see if there is any benefit of this vaccine-based approach in multiple myeloma. Now next, I want to switch gears and talk about the the major attraction of the immunotherapy right now, the cellular therapy approach. So the cartoon here gives how the cellular therapy is given to the patient. But if you look at the history of the cellular therapy in myeloma, the first cellular therapy was with the use of allogeneic stem cell transplant and the donor lymphocyte infusions. Now we have several engineered cell therapy approaches, in particular the chimeric antigen receptor T cell therapy, which is showing a lot of promise in multiple myeloma. There are non-T cell approaches like NK CARs are being developed, and there are T cell receptor-based therapy as well in myeloma. But in particular, our discussion today will focus on the BCMA-based CAR T cell therapy because the data we have and the several other trials that is exploring that approach. The main side effect, as in lymphoma CARs, were safety with the CRS and neurotoxicity, but definitely less incidence than the lymphoma CARs. And the early results that we have look very, very promising. Actually, the FDA led to the approval of the first CAR T cell in myeloma called Is Bcma, or Idecaptogen Viclucel, from the BMS, which is approved like 2 weeks ago. And soon we're going to be approved being another construct from Janssen, called Cilta-cel or Cilta-Captogen Autoleucel. So a few kind of slides on that. As I said, the BCMA is the target for this CAR T as well. This is the first phase 1 study of BB2121, now we call ID-cell, that was presented and published in New England Journal of Medicine in 2019. It's a phase 1 dose escalation and dose expansion study of 33 patients. The dose in escalation, the dose range tried were 50 million cells up to 800 million cells, and the expansion was 150 to 450 million cells. This is the data. As you can see, the key data, the median age was 60. The median prior lines was much higher, 7, and the range is up to 23 lines. So very heavily treated, no doubt about that. The quad refractory is about 18%, means refractory to both PIs and both IMiDs. And this is some of the efficacy data. The 85% response rate, definitely unprecedented for the patient population they chose. The PFS was 11.8 months because based on the Mammoth study, which is the study that looked at the patients who relapsed after CD38 monoclonal antibody, the median PFS and survival was not, the median PFS was about less than 5 months, whereas the PFS here is about 12 months. And this is some of the side effects. About 76% CRS, grade 3 was much lower, 6%, neurotoxicity was 42%, and the grade 4 was 3%. And this is the ongoing responses that is present in those patients. As you can see, patients below 150 million, right? 50 million, the patients who received a 50 million dose didn't have any responses. So the objective responses were only seen in patients over 150 million. That's why that led to in the expansion phase, the dose tried were more than 150 million only. Now, because of this very exciting data on the phase 1 led to the design of this pivotal study called KARMA, the phase 2 pivotal KARMA study. And this is the design, patients with more than 3 prior lines of therapy, exposed to all 3 class agents, in particular, IMiD, PI, and the CD38 antibody. And the design is they go to leukapheresis and lymphodepletion with the flu-cy, the standard conditioning that is used. And about 158 patients were screened. About 128 patients were dosed with this construct. The primary endpoint was overall response rate, and there is some of the secondary endpoints. So this is some of the key efficacy and safety data on the left-hand side. As you can see, about 61 is the median age. The high-risk cytogenetics was present in 35%. Extramedullary disease was present in 39%. The median prior lines is similar to as the phase 1, 6, with the range up to 16. And triple refractory to both PI, IMiD, and CD38 was 84%. So in general, as with the phase 1, very heavily treated. And this is the 3 different doses were tried in that, 150, 300, and 450. The 450 is their the recommended phase 2 dose, that is the dose that is being used, it's being approved. And as you can see, the overall response rate for everybody was 73%. And there were MRD negative CR in all those levels, but in particular, overall, about 26% of MRD negative CR, and about 39% were with the MRD and more than VGPR. And this is the safety data. So the cytokine release syndrome and ICANS, also known as immune effector cell associated neurotoxicity syndrome, which is the 2 main side effects that is kind of worrisome for these patients. And this is at different dose levels, but you can see the grade 3 is much lower, about 5%. And the median onset of this CRS was very interesting. It was about very quickly, 1 day, and the range of 12 days, and lasted for about 5 days. And the grade 3 ICANS was about 3%, and the median onset was in 2 days. And this is some more efficacy data in terms of the follow-up. As you can see, the PFS was about 8.8 months, so slightly low than what they saw in the phase 1 study. And the overall survival was 19.4 months. Definitely very, very exciting and unprecedented for this kind of patients to have this overall survival. But what is important is looking at the duration of responses. You know, for patients who have a CR and stringent CR, for those patients with the deep remission after the CAR T, their duration of responses was 19 months, and the PFS was about 20 months, and overall survival for overall patients was about 19 months. So this is very exciting. This construct is approved now, and we are started using this construct in for our patients as well, which is approved as patients with 4 or more lines of therapy, so kind of heavily treated and the kind of end stage patients who have no other options left. This construct is being used in different settings in the earlier lines, in patients with the early relapse, and also patients with newly diagnosed high-risk patients. The trial is called Pharmafer trial, and we have a trial that is open right now, which we are enrolling for that group of patients as well. So we have to see how they pans out when compared to the standard therapy that we have in myeloma, in terms of both efficacy and duration of response and safety. The other construct, which is a very exciting construct, is cilta-captazine autoleucel. This is, if you recall, this is the construct from Legend from China, which they presented data in ASCO in 2018. Very exciting data of 100% response rate in these patients. So this is a little bit different structurally. If you look at it, there is a biparatopic binding. So there is a 2 Bcma target single domain antibodies designed to confer affinity. But the co-stimulating domain is pretty much the same. Is a 4-1BB for both this one and the other construct that we talked about, and the phase, the Legend construct, which is the same construct, showed very powerful responses in heavily treated patients. So that the US study was done in the name of CARTITUDE 1 and 2, and this is the design of the study. The eligibility criteria is pretty much the same as what it was in before the prior CAR T, more than 3 prior lines, and the dose here is much low dose as compared to the ID cell, which is about 0.71 million cells per kg on average. And again, the conditioning is flu-cy conditioning, lymphodepletion, and followed by the CAR T infusion, and looking for the both safety and efficacy. So this is the key efficacy and safety data. Very impressive efficacy data. About 97 patients were enrolled and 97% response rate. They're also very heavily treated. Median prior lines were 6, same as the other construct. Triple refractory rate 87.6%, and they also allowed the prior allogeneic transplant patient. Actually, our center enrolled patients who had prior allogeneic transplant, and so they were able to get the very good responses even after allogeneic transplant relapse as well. And this is the MRD and the CR, the stringent complete response data. Again, very good at this stage with the overall response rate of 93% with MRD negativity. And then all treated patients, if you look at, there were 55% with MRD negativity. So this is has been filed for approval. So it's most likely it's going to get approved sometime in the fall of this year. And so we'll have 2, kind of a construct for myeloma CAR T by the end of this year. Now, again, the one thing we need to talk about is the safety of this construct is a little bit different than what we saw in ID cell. So there were the grade 3 CRS was about 5%, pretty much same as the other one. The median time to CRS was a little bit different. If you look at it, the median time to CRS was within a week, as compared to the other construct, which is like right away after the T cell infusion. Here is about a week, and the duration is pretty much the same. But interestingly, there is some interesting neurotoxicity that happened in this construct, in particular delayed neurotoxicity. For example, the grade 3 ICANS, which is related to the T cell, is about 2.1%, and median time is similar right after the CRS. But other neurotoxicities, about 9.3% of the patients, almost 10% of patients had other neurotoxicities, in particular toxicities like Parkinsonian-like feature and movement disorders and so on and so forth. They happen pretty late. If you look at it, the median time to onset was 27 days, quite late after the than compared to the other ones, and also with compared to the ICANS. So we don't know why this kind of toxicities happen. We have a trial right now looking at the same construct in early lines after 1 to 3 prior lines, and we have dosed this construct for about 7 patients already, and we haven't seen that delayed neurotoxicity. And from what I know is in the early line trial, they have not shown any kind of delayed neurotoxicity. So it will be interesting to see if that if once you enroll all the patients, we see this delayed neurotoxicity. But at the same time, the ICANS, the grade 3 ICANS is pretty comparable with the other construct. And this is the median PFS. The median PFS has not been reached, and these are some of the total out disposition of the patient. Deaths due to adverse event and deaths due to adverse event related to treatment and are not related to treatment. As you can see, about 14 deaths out of that 5 had a progressive disease, and 6 deaths were due to adverse event. In particular, the neurotoxicity led to the deaths of these patients. But the overall efficacy-wise, very powerful, very promising, and the median PFS as of the last data point has not been reached. I think this is already coming out in publication soon, and we'll see the details of this, the efficacy and safety in the paper. Now, this is a comparative analysis of this 2 construct, and in addition, there is another construct, Orva-cell construct, which is from the Juno or the BMS. Unfortunately, that is not going further for the future development, but this is some of the data that we can just look for academic interest. Overall, they all are very powerful. The overall response rate for heavily treated patients ranges from 73% to 97%. The Cellular is the winner with a 97% response rate. Very deep responses, including the MRD negativity, and the median PFS, as you can see, 8.8 months for the ID cell, which is from the BMS, and not reached for both the Cellular cell and Orva-cell construct. Now, there is explosion of the CAR T cell in myeloma, and now the CAR T is being used in different settings, trying to target the several limitations that we have in myeloma. And for example, they are being used in early lines, they are being used in combination approaches, they are being used with different memory functions, the composition of the T cells, like CD4, CD8 composition, used with just mRNA CAR, different construct, and different other targets. In addition to BCMA, there's a target against CD38, there's a target against APRIL, which is the ligand for the BCMA. There are targets in CS1, which is that, and there is even the donor. There is a donor CAR T, like allogeneic CAR T, is being developed, and so on and so forth. So it's very exciting time for myeloma CAR space. Of course, we have to see. It's too early to say the definite role of this kind of approach that takes place and that kind of plays in myeloma, but we know for sure that this is slowly going to be a kind of the kind of central stage in multiple myeloma, in either by itself or in combination. Despite all this the impressive efficacy and the promises that the immunotherapy in particular has, there are some challenges as well. So what are the major challenges? I'm going to talk in the next 10-15 minutes about the major challenges of myeloma immunotherapy in particular. We know in our clinical practice that even though it holds a lot of promise, there is quite a heterogeneity in terms of responses. Not all patients respond well, and some respond for a very short time and some don't. That is one challenge. The other challenge is we talked about is safety and toxicity, in particular with the CAR T cell. And last is timing. When is the best time to use immunotherapy in myeloma, and what is the sequencing, and what is the combination, the best combination? And for all this, we don't have any biomarker. We don't have any immune biomarker to predict the safety, to predict the responses, the efficacy, and so on and so forth. So that is an unmet need to have immune biomarker for all of these immunotherapeutic approaches. So we are kind of interested in this one particular target as a potential immune regulator in multiple myeloma. So in myeloma, we know that it's a niche-dependent disease. And the niche is mainly in the canonical niche, or what you call as canonical niche, is mainly composed of bone supporting elements like osteoclast, osteoblast, osteocytes, and they provide both trophic and regulatory support to the myeloma microenvironment. But at the same time, we know that myeloma can survive, in particular now, as extramedullary disease, and even extramedullary plasmacytoma is not uncommon. So the non-canonical niche is also possible for the myeloma to survive. So what you can see is that the basic elements of extracellular matrix and stromal cells, can be a common denominator besides the bone supporting elements. So extracellular matrix, stromal fibroblast, and immune cells. And in that extracellular matrix, there are several matrikines that are responsible, and that is being investigated as a potential immune-regulated myeloma. They are either biglycan, proteoglycan, collagen, hyaluronan, and all those stuff. So I'm working with the Fortis in one particular extracellular matrix or matrikine called versican. So the versican is actually is been implicated in several other cancers and metastases that helped in helping kind of a progression of the disease, and also in kind of a metastasis of the disease. What we know in myeloma is that the Fortis group kind of in a preclinical model, what they found out is that the versican can deposit in around myeloma lesions and can help in immune regulation of the microenvironment. And in the human myeloma cell lines and also in the preclinical model, what they found out is that this versican can be degraded by a very regulated proteolysis called ADAMTS proteases and release a daughter molecule called versicine, which is a kind of a breakage at the one glutamine and alanine kind of amino acid path, and that lead to the product called versicine. And what they found out is that versicine, so when this versican, in the presence of versican, the microenvironment was very tolerogenic. But when the versicine kind of gets produced from the degradation of the versican, that can lead to the recruitment of the T cells, and also reversing the dysfunctional dendritic cells, and that can be immunogenic. So these all T cells can migrate in the presence of versican and can be immunogenic. And so it also helps in the maturation of the APC, and also helps in trafficking of the tumor and the CD8 cells to the tumor bed. So suggesting that there is a potential role of exploring this extracellular matrix as a potential immune biomarker in myeloma for all sorts of immunotherapy. So what we did was we tried to explore that, whether this can be used as a novel immune biomarker in multiple myeloma. So we use this versican staining, which can be done by immunohistochemistry, and in patients who are after transplant at day 100 when they recover, and they looked at the VCAN proteolysis, and that means measuring the versicine, which is a daughter molecule, at different stages. So we found 3 different groups: low group, moderate group, and high group. And in that, we also consequently stained for the CD8 T cells, and we also found that the more the versican proteolysis or the VCAN, you can see the higher number of the CD8 T cells stained in that group. And this is some of the this is the 35 patient study, and we have now updated data at last year ASH with about 60 patients. And this is as you can see some of the demographics here. The conditioning, all these patients went to transplant the MEL 200. They were about 70% more than very good partial response rate. About 51% had low VCAN proteolysis, and the 49% had very high VCAN proteolysis. Paradoxically, what we saw was that we definitely did demonstrate that this VCAN proteolysis was correlated with the highest CD8 burden, which is what that was observed in the preclinical model. But what we found out that the patient who had the high VCAN proteolysis had also worse outcome, which is kind of a paradoxical observation than what we saw. So this is a summary, and there is ongoing work right now, which we are exploring in different settings. But this is what the summary we have for the VCAN or the VCAN proteolysis. VCAN accumulates in myeloma tumors and can help in the immune regulation of the myeloma microenvironment, where the ADAMTS proteolysis of the VCAN can help in the generation of the versicine, and the proteolysis generates the CD8 T plus CD8 T cells infiltration at the tumor beds. But there is a paradoxical association of weekend proteolysis with dismal outcomes. So next steps, what we are doing right now with the use of single cell sequencing and the nanostring analysis, we are looking at those functional characteristics of those T cells. Are they senescent T cells? Are they exhausted T cells? Can we use this weekend proteolysis as a guide for intervention in these patients? And the trial that I mentioned to you about the vaccine trial, we are using the weekend proteolysis as a correlative biomarker to see if the patients who receive a vaccine after transplant, we can have a predictive biomarker or prognostic biomarker using weekend as a part of the biomarker. So that all these results are we are testing right now, and hopefully we'll have the results by the end of the year or sometime next year. The next challenge is the timing of immunotherapy. So what is the best timing of immunotherapy is an ongoing question. A lot of people kind of argue that potentially the post-transplant period is the best timing for immunotherapy because of several reasons. In particular, there is a reduced tumor burden, and therefore there is fewer immune targets and the reduction of the tumor-mediated immune suppression. In particular, the decreased number of the T regulatory cells, myeloid-derived suppressor cell, and so on and so forth. So all these studies right now are targeting the post-transplant phase as a good platform. Having said that, we need to kind of do more studies, and there more studies needs to be performed in that setting to see what is the best time and that the patient can achieve the best response to the immunotherapy. For example, the vaccine is used after post-transplant, but a lot of CAR T cells are being used in a relapsed refractory setting, and when they are using in the front line, they are used at the different stages. But with this evolving data, we'll find out what is the best time of immunotherapy in myeloma patients. And the other modification that is happening is using a safer CAR T strategies. One of them is, as seen here, is an mRNA CAR instead of the DNA CAR. So the mRNA, as compared to the DNA, has a very transient expression, and they do the job and go away. Actually, we have a clinical trial in myeloma, which I'm the local PI here for the mRNA CAR T in myeloma. We are just starting enrolling patients in high risk upfront, myeloma after transplant for the mRNA CAR. We know one thing for sure is it's very safe with no, any higher evidence of neurotoxicity and the CRS. The other one is using on and off suicide gene or the CD20 suicide gene to switch on and off the T cells, kind of regulate the T cell activity and also diminish the the side effects. So these trials are ongoing right now. And so this is one step that we can hopefully get some safer CAR T construct in the future. And this is the other trial that I want to just briefly discuss about, this resistant T cell therapy trial, also called as RAPA T cell therapy trial, that we are enrolling right now. This is non-engineered T cell approaches where the T cells are grown ex vivo in the setting of interleukin-2, interferon gamma, and high-dose rapamycin. Rapamycin is, as you know, it's an inhibitor of the mTOR pathway, which is used by the T cells for survival. So the idea is that if you block that pathway and then polarize these T cells in the presence of cytokines, then you can get the more Th1 responses. So this is in collaboration with the Dan Fowler, who was in NIH before. So this is what they saw in the pre-T cells and post-T cells growth. There are more polarized to Th1 expression. We are currently enrolling patients right now in this study. It's a phase 1-2 study. And we are seeing some exciting results, and we'll see, the study probably will finish accrual by the end of the year, and we'll have some results to share either, that sometime next ASCO. So this is another non-engineered T cell approach that is being explored in myeloma as well. And we have to see the role and the kind of position of this kind of non-engineered approach and as compared to the engineered T cell approaches. So in conclusions, I can say that myeloma immunotherapy is here to stay. There is excellent activity of monoclonal antibodies in relapsed and newly diagnosed setting. Very impressive early results with BiTEs, ADCs, and CAR T and in relapse setting, and it's moving forward in the early lines and also newly diagnosed setting. There is a potential for cure with the combinatorial immune approaches, but the challenges are we need a robust translational work for immune biomarker, which is an unmet need in myeloma for all immunotherapy. We don't know that optimal timing of this immunotherapy, what is the best sequencing, and what is the best partner for this immunotherapy. Further research are needed to understand the immune microenvironment rheostat or immunostat that happens in myeloma, and very profound immunosuppressive status that happens in myeloma. So further work is needed to understand the tumor microenvironment, to redress the balance of the immunosuppressive pathways. And last but not the least, we need this multi-center collaborations with both clinical investigators and basic sciences for safe and effective immune approaches. And that's all I have. I want to thank my team, which at Medical College of Wisconsin, both clinical and translational team, the basic science team, and the collaborators that we have. And thank you for your attention.